Panagiotis. Okay, got it.
Thanks a lot from Professor Choli for the nice introduction.
Louder.
Please. Fix the sounds. It's in the mute.
Okay.
It's muted.
Yes.
So hello everyone from me. I'm Panagiotis, and I will share with you how we use mRNA technology to manufacture the way to approach ORF3A protein secrets. So it's a really recent project that we are working on, and one of the two projects that I will present you, and it's about the mRNA technology. So there are 5 seemingly easy steps to master mRNA manufacturing, but as a molecular technique, it contains a lot of hindered troubleshooting and a lot of design behind each step. The first step is a more computer-based step that requires the design and optimization of the nucleotide sequence of the protein that we want to study. So we need to recruit bioinformatic tools and databases so we have the optimal design. We must take into consideration two essential factors for this, one of which are if we want to study the wild type protein or the mutant protein, so we design the sequence that will contain the mutation that we will study, the mutation of interest. And also we must take into consideration of optimizing the codon sequence that will be translated to the respective amino acids according to the host cells that we will use later in the mRNA transfection step. So we must take on consideration if there will be mouse cell lines or in vivo in mice, or there will be transfection in human derived cell lines. Then we proceed with the conventional cloning into a plasmid vector. The important in this step is that the plasmid vector should contain a specific promoter according to the in vitro transcription kits. Kits of in vitro transcription are commercially available and usually based on the activity of sp6, T7 or T3 RNA polymerase. So our plasmid vector should contain one of these 3 promoters. Of course, today there are many companies that are specialized in DNA synthesizing, so we can surpass this step by just designing the nucleotide sequencing and sending it to the incorporating industry to design and insert our gene of interest into the plasmid. Then important step is the linearization of the plasmid vectors. This step is really important in order to generate mRNA of specific length. Otherwise, RNA polymerase will start from the promoter and will go round and round to plasmid vector, generating mRNAs of vary lengths. So the linearization step is a must. And then we proceed with the in vitro transcription reaction. There are many commercial kits, as I said, where we can generate the mRNA that we can either use it as it is and purify after purifying, or we can add co-transcriptional modifications such as the 5' cap or the 3' poly-A tail to enhance mRNA stability and protein translation efficiency later in the mRNA transfection step. So if we are doing an mRNA manufacturing in a small scale, there is also lipid nanoparticles commercially available that we can encapsulate our mRNA and transfect the cells. Otherwise, for example, in pharmaceutical industry, there is a 4 0.5 step, if we can say so, that we must design a proper lipid nanoparticle or a nanoparticle that is biocompatible and non-toxic in order to proceed with the mRNA transfection. So the last step is the mRNA transfection, where the mRNA is delivered into the cells and the protein of interest is translated. So in this project, we examined the ORF3A protein of SARS-CoV-2, a viroporin that forms homodimers inside the cells, and it really is an important protein for viral pathogenicity and release, as well as for many signaling pathways, such as apoptosis, inflammatory response, and autophagy. But until 1 year before, until 2023, there was not a known mechanism about how this protein is involved in these signaling cascades. So, an important signaling pathway that we want to study was the unfolded protein response. Unfolded protein response is a modulator of cell homeostasis where there is a disruption between the capability of cells to express proteins and to fold them. So there is a condition triggered called ER stress, and host cells try to relieve this ER stress by activating 3 transducers, ATF6, perkinase, and TYR1 elements. Misfolded proteins accumulates and binds to these proteins, thus activating them. And IRA1 and ATF6 activate specific gene expression, such as redox enzyme expression and saperonins expression to relieve ER stress. However, PERK have a lot different pathway where it phosphorylated eukaryotic initiation factor 2A to switch off the general mechanism of translation in order to reduce the protein folding load in the R lumen. So the aim of the study was to use mRNA technology to access the virulent nature of ORF3A. The role of perkinase, as it was the last, we didn't know as much as the other 2 transducers, ATF6 and TYR1, and its role, PERK's role in ORF3 transfected cells. And then if we can use some inhibitors, PERK inhibitors as potential therapeutic approach. So in our project, as I said, we did an mRNA manufacturing in a small scale, where we cloned ORF3A gene after designing and delivered it in a plasmid containing a CMV promoter. So we had to clone it in a T7 promoter, in a vector containing a T7 promoter, as well as we cloned GFP in another PT7.7 vector, which is a lot different from PT7 iris HCDNA vector in which ORF3A cloned. PT7 iris HCDNA contains the T7 promoter, which is essential for the in vitro transcription reaction step, also contains an EMCV iris element, namely an internal ribosome entry site that enhances the protein translation efficiency later in the mRNA transfection step. And there is also the capability of adding a poly-A tail in the 3' of the generated mRNA. However, PT7.7 only contains the T7 promoter. It doesn't contain the iris or the poly-A characteristics, just the T7 and the histag fusion histag in order to detect later our protein inside the cells. So we proceeded with the linearization downstream of T7 promoter and gene of interest region. The in vitro transcription reaction was held with co-transcriptional modification during the reaction, where a 5' cap was added. We used anti-reverse cap analog, or ARCA, which, although it gives less mRNA efficiency during the in vitro transcription reaction, but ARCA has the has the capability of binding to the 5' region of the mRNA only the correct orientation, contrary to other kappa analogs, which can also bind in an adjacent orientation. So ARCA also can enhance the mRNA stability more efficiently than other caps. And also we added in the in vitro transcription reaction a poly-A tail in the 3' of the mRNA. We then purified the mRNA, and after DNA treatment to get rid of DNA traces, we collected the purified in vitro transcribed mRNA. An important reason why we used mRNA transfection was to simulate the way in which the virus enters the host cells, as SARS-CoV contains a positive single-strand RNA, which is similar to the host cells mRNAs. So we continued with the transient transfection step of A549 lung epithelial cancer cells with the mRNAs. We can see that the mRNA was successful both with using real-time PCR and Western blot techniques. We can see that at 48 hours post-transfection, there is a peak in the mRNA levels of ORF3A, and the peak for the protein expression levels are 96 hours post-transfection. For GFP, similarly, we can see that the maximum peak of protein expression are at 96 hours post-transfection, and also validated by confocal microscopy images at capture, where we can see the diffuse localization of GFP into the cells. So these clues give us that the mRNA transfection was held successfully. Then we went deeper in the ORF3A functions, and we found that ORF3A functions as an ER stressor, can induce ER stress, as can in the ORF3A transfected group, we can see the upregulation of PERK, ATF6, and XBP1S, an executioner of IRA1 pathway. So ORF3A can activate all three branches of UPR, but there was a notable overexpression in PERK. There is a notable PERK overexpression in the ORF3A group. So we also studied some executioners of these pathways, such as the phosphorylated eukaryotic initiation factor and CHOP, which were also upregulated. GFP, I should highlight that GFP was also used in order to exclude possible artifacts that could insert during the mRNA transfection, and that all these observations are not due to the mRNA transfections, because there was the probability of the lipid or the buffers used could be potential ER stressors. So we found out that GFP mRNA transfected cells showed non-significant difference compared to control cells. So this inducing of ER stress is ORF3A specific. Then, in order to unveil the ORF3A nature and the interconnection with the PERK kinase, we treated transfected cells with an inhibitor of PERK kinase, GSK 2606414 or GSK 414. We found after an MTT cell viability assay that the optimal concentration for treating cells are 1 micromolar of GSK. So we then proceeded with the same logic as previous by transfecting cells and then treating them with the GSK for 48 hours for the total of 96 hours post-transfection. We found out that the treatment with the GSK reduced the expression of PERK executioners, but did not affect the other UPR branches. Then we showed a lot of reports that PERK is a homeostasis modulator and is involved in many cellular processes such as apoptosis. So we wanted to see if ORF3 induces apoptosis via the PERK pathway. First of all, we found out that ORF3A induces apoptosis by activating caspases and PARP1, and we can also see the mRNA overexpression of BCL2 family protein, apoptotic family proteins, and the regulation of BCL2. In the case of the treatment with the GSK, we found that the caspases are retained in the precursor form, so they are not activated. And we also observed an upregulation of BCL2 both in mRNA and protein levels. In order to further validate the results of the GSK experiments, we also used an pan-caspase inhibitor, which is known that inhibits apoptosis by inhibiting caspases. So we used ZVAD in a molecular concentration of 50 micromolars. And indeed, we saw that also treatment with ZVAD can favor cell survival. So the mechanism of ORF3A apoptosis is shown in this slide. ORF3A activates PERK. The activated PERK then phosphorylates EIF2A, ATF4, and then the pro-apoptotic transcription factor CHOP, which then activates caspases and apoptotic members of the BCL2 and BH3 only proteins families. So thus cells are driven to apoptosis. But there are two cases where we can inhibit apoptosis in later steps with ZVAD just by inactivating caspases, but we can go one step earlier where GSK treatment with GSK can inactivated PERK. So PERK is in the inactivated form, so we cannot see this upregulation of CHOP and thus the activation of the apoptotic biomarkers such as caspases and BCL2 family proteins. So by treating cells with GSK, we inhibited ORF3A-induced apoptosis. Then another function of ORF3A that is related with ORF3A, this is the inflammatory response of the cells. Indeed, ORF3A can upregulate many pro-inflammatory factors such as interleukins 1b6, 8, and 18, as well as the active phosphorylated form of NFKB, and can induce the phosphorylation of the inhibitor of NFKB, IKBA, which phosphorylation leads to its degradation. Then we treated cells with transfected cells with GSK, and we found out that PERK inhibition restrained the expression both of intracellular and extracellular pro-inflammatory factors, such as the major interleukin 1b, which is shown here, the interleukin 18, 6, and 8. Also, there is a regulation in the phosphorylation of the P65 subunit, active subunit of NFKB, and down regulation of the phosphorylation of IKBA, thus indicating a less active NFKB. In order to discriminate and to show that apoptosis and inflammatory response are not correlated, we also used the ZVAD inhibitor. So we treated cells also with ZVAD. This treatment didn't affect the levels of both the pro-inflammatory factors and from the phospho P65 and IKBA. So ORF3 induces the inflammatory response via the PERK-NFKB pathway, as shown here. After the transfection, ORF3 activates PERK. Activated PERK phosphorylates EIF2A, which inhibits the general mechanism of translation, reducing the expression of IKBA, thus leading to its phosphorylation and degradation. In the same time, there is for phosphorylation of the active subunit of NFKB, which translocates to the nucleus, where it can express the inflammatory cytosines. So in this project, we successfully used mRNA technology and mRNA manufacturing to show that PERK could serve as potential target for developing novel therapeutic approaches against COVID-19, as the lipid ORF3 mRNA complex was introduced to cells where ORF3 was translated, and then ORF3A activated PERK, and activated PERK drive cells to apoptosis and their inflammatory response. When there was the treatment with the GSK 414, PERK retained in the inactive form, thus inhibiting apoptosis inflammations and favoring cell survival. So I want to thank my professor, Theodora Choli-Papadopoulou, Dr. Eleni Papachristou, who is involved in this project, and other members of the Professor Choli's team. If you want more info about about this technique, how we used it, how we solved many troubleshooting steps according to the designing or during the mRNA, the in vitro transcription step, or about knowing about the commercial kits that are available, you can contact either me in pankerdim@chemaut.gr or Professor Scholi's email. And I want to quote that the mRNA technology could help us unlock the secrets of many proteins and could be the future of treating many diseases. So thanks all of you for your attention.
Thank you, Panagiotis. I don't know, Nikos, if you think that we can have questions on this topic, and after that we can present the next presentation.
I think this is more appropriate. I think it's better.
Huh?
Therefore, yeah. I think this is better to if there are any questions for this part of the presentation to Panagiotis, please, you can ask now.
Okay.
Any questions? I can hear. No. I could ask a brief question regarding the selection of the cell line. Can you hear me?
A little bit louder.
It's a bit silent.
It's better now.
A little bit, okay.
Okay. About the selection, first of all, it is a really interesting and hot topic, and thanks a lot for sharing this work and the perspectives of this. As for the cell line that you selected, of course, a cell line, a lung cancer cell line, a cell line of lung cancer origin. Okay.
Yeah.
Yeah. Would you think to try some of these approaches, let's say in a lung fibroblast or a non-neoplastic cancer cell line, as for to compare and study this approach in a more general approach? What do you think?
Yes, most of the one part of this work were about master thesis, and now as a PhD student, I will continue by introducing the mRNA into the cells of normal lung cells, if we can say so, normal lung cells, and fibroblasts, maybe in fibroblasts, in order to see different cell signaling pathways, as there may be a connection of ORF3A with fibrosis.
Actually, we used not only the cancer lung cells, but also the MRC5, which are immortalized lung epithelial cells, and usually the colleges in the world, so they use that as normal, in parentheses, epithelial cells. Okay, mean these are immortalized. Okay. Actually, now we are on the way to investigate also the plotting the ORF3A transfecting the CACO3 cells from the gastrointestinal part of our organism. And actually, we had very nice and very important results, but at the moment we have to work on them, because we can see that the protein enters also the nucleus there. So it's not only, the same trafficking, cell traffic as in the lung cells, so it's a little bit different. And we are now on the way.
Είχε κλειστό το μικρόφωνο.
I had that.
So Professor Choli was saying that we started seeing some results in MRC5 cells, which mostly consider a normal fibroblast lung cell line. And we also studied, we have some starting results, early results in CACO2 cells from gastrointestinal site. I think it's colon cancer cells. Where ORF3A exhibited a total different localization, where we saw that ORF3A could enter and the nucleus of the CACO2 cells. In A549 cells, we showed that we found out that ORF3A resides mostly in the cytoplasm. So there are many steps in this project to proceed, such as showing the more general behavior of this protein both in cancer and normal cells and in other cells, also in macrophages for the inflammatory response. So we have reached now to master this mRNA technology, as I said. So we proceed depending on what we want to see and in what cell line we want to study it.
Okay.
So if the people do not have other questions, probably we can go on to the next presentation, or what do you think? I mean, we can see also the cameras from the others in the cameras on the picture.
No, I think it's better to move on because time flies. So,
okay, we can go on the second presentation.
The one that should be that or expected presented by Eleni.
Yes.
Okay.
Unfortunately. Okay. So the next one. So these are the mRNA vaccines now. Okay. Actually, these results are not published. until now. We worked on that in the frame of a proposal, and therefore we cannot give you a lot of details.
Do you want me to stop recording this part? Because we can do that too, if you want.
I think it will be better. Yes,
Okay.
Adolfo. Thank you.
That's
Adolfo.
good, no problem.
Thanks a lot.
Okay, thank you.